Welcome to Let's Talk Micro.
And I just want to start by thanking you all for listening to the last episode, episode zero on introduction. I am so happy to be here talking to all of you, to once again, be sharing my knowledge, my passion. We go through the show, I'll be using some technical terms, so I'll be referencing to a textbook. There are some really good ones out there. I use Bailey and Scott's microbiology. And once again, a quick disclaimer. They have no affiliation. They're not sponsoring this podcast. I just think it's a great reference book, so I'll be using it. And so last week, we talked about the role of technologists or medical lab scientists and what we do and how that work impacts healthcare. Like I mentioned, you have an infection, you have a cut, etc., and you go to a clinical, you go to a hospital, right? So what does the doctor do? He collects a sample. And then that sample is sent to the lab, and here's where we come in. What do we do with it? Right? So the doctor orders a culture. And like the word says, we are cultivating the sample. We are trying to see if something grows out of it. And in this media, or agar, there are nutrients that the organisms need to grow. Just like us. We need food. So they need food also. They need some nutrients. And there are several types of media. Right? So on this podcast, I am focusing on media for bacterial cultures. Normally, the media is classified based on its functions and what it contains. So, and this here's where we get a little technical. So, basically, in bacteriology, we have four categories of media. We have enriched media, which has specific nutrients required for certain pathogens. A great example you have like limb broth, which we use it for the recovery of group B strep. We have nutritive media, which contains nutrients that support growth of most organisms. So basically, it grows pretty much everything. And a great example of this media is blood agar. Every student in basic micro, when you get started in the lab, you work with blood agar, right? So when I'm talking about blood agar, everyone knows what I'm saying, basically, right? Um, it has like sheep red blood cells. And it is definitely one of the main agars that we use in cultures. I will say that almost every culture panel includes blood agar. And then we also have selective agar and we have differential agar. And selective agar is just, it has one or more agents that inhibit the growth of certain organisms while selecting others. That's just what it is. Two classic examples that we have PEA and we have MacConkey, right? MacConkey has those bile salts that inhibit the growth of gram-positive organisms. So it selects for gram-negative rods. And another great example of selective agar is PEA, phenyl ethyl alcohol agar. So it is used for the selection of aerobic gram-positive cocci and bacilli. So that is not to say that other organisms will grow, but maybe somewhere down the line we'll talk about that. But these are two great examples. They are included on your basic battery of cultures, and I will talk more about this. But definitely data on blood, PEA, MacConkey. And, here's for your students out there, here's where you have to be careful. You have to be as specific as you can be when you're describing, the purpose of this agar. These are very typical questions that get asked. Maybe if you're doing a clinical rotation at a hospital, if you're taking a micro class. So what I mean, for example, if I give you a test and I say, what kind of media is MacConkey, right? And your answer is selects for gram negative. Okay. So gram negative one, you see, by leaving it out like that, you are telling me that I can go ahead and I can put an Neisseria gonorrhea, a Pastorella, a Haemophilus, and it will actually grow. And as you know, that's not true. And maybe some of you are there, some micro enthusiasts are saying, well, if I put a Pastorella rogenes, it will grow. And for that, you will be right. But overall, the other pastorellas, like Pastorella canis, Pastorella multocida, they're not going to grow in MacConkey. Pastorella aerogenes is an exception. And I have a great story about Pastorella aerogenes. And so this happened a few years ago. I was working in bacteriology. And normally when you start seeing pastorellas, you get your classics. Canis and multocida. And what's pastorella? It's a gram-negative coccobacilli that is normal flora in animals. Multocida can be normal flora in cats and dogs. Canis is for dogs. And there's another pastorella, which is Pastorella aerogenes. So I worked this culture, and this was before the Molotov days. For those of you that know the Molotov, you get an idea really quick. My hospital at that time didn't have one, so we were still doing the Vitek. So I go ahead and I'm working my gram-negative rod. It's growing on Mac. I set up a Vitek, and then the next day, this is the wound bench. There's not a 24-hour read, so basically you work it up, and then the next day you find out what it is. So I come back the next day, and then I'm looking at my ID, and it says Pastorella aerogenes. So I'm like, what? Like Pastorella aerogenes. I was a little surprised. Pastorella not growing on Mac. So here is where I go to another great resource that has no affiliation to this podcast. I go to an ASM book, American Society of Microbiology. They actually establish the procedures on how to, what media we use, how to properly work up the cultures, basically what we need to rule out on a particular source. And as I look information on this organism, I look Pastorella aerogenes. It grows on MAC, gram-negative, coccobacilli, biochemicals match, and everything. And it says it is normal flora in pigs. So it is caused by pig bites. So I go ahead and I decided to do a little research, and it turns out that yes, my patient had been bitten by a pig. So I was a little, as a micro enthusiast, like I was really excited, and that was the first time and the last actually that I had one. But with this, circling back to your selected media, you have to be specific. Like I said, if you're answering what MacConkey is for, select it for gram-negative rods. So you need to be specific because there are gram-negative rods like Haemophilus or anaerobes that do not grow on MacConkey. And you can actually lose points for that in your test. And not only that, you will be actually telling wrong answer. So just be careful when you're going over these. And then we have differential media, which has ingredients that basically they allow some colonies to exhibit some characteristics that makes them different from other colonies growing on the same plate. We have two great examples: blood agar, which is differential for hemolysis, right? And what is hemolysis, right? It's a lysis of red blood cells. And there are three types of hemolysis. You have alpha, you have beta, and you have gamma hemolysis. So alpha hemolysis is the partial lysis of the red blood cells in the agar, right? So it gives you a green color, greenish. Beta is complete lysis, so it makes you look clear. And then you have gamma, which is no hemolysis, so it stays the same. And another example is MacConkey, and it's differential for lactose fermentation. So organisms that ferment lactose, right, they turn pink. And an example of enriched media, besides the limb broth, is chocolate agar. And chocolate agar is basically heated blood agar, and then it releases so then it makes both. It has both the X and V factors, which Haemophilus, like Haemophilus influenza, needs to grow. So that's why Haemophilus can grow in chocolate, but it cannot grow in blood agar. Right? Blood agar has the X factor, but doesn't have the V factor. So that's what chocolate Chocolate has both of them. And it also helps, organisms like Neisseria gonorrhea to grow better. And that brings us to the quiz time. So let's start with blood agar. What kind of media is blood agar? It is nutritive and it is differential for hemolysis. It is not selective. Gram-positive cocci, gram-positive rods, gram-negative rods, gram-negative coccobacilli, yeast, they will grow on this agar. So it is not selective, but it is differential for hemolysis, right? This is when you can see that nice beta hemolysis of the group B, group A, beta hemolytic strips. That hemolysis that Staph aureus produces. You can see them. You can see beautiful examples in this agar. So it is differential, and it is nutritive. And what kind of media is MacConkey? Well, MacConkey is selective and it is differential. It is selective for gram-negative rods. Remember that distinction, not gram-negative. Gram-negative rods. So it is not gram-negative. No, it is not selective for gram-negative. Neisseria do not grow on it. Anaerobes do not grow on it. Haemophilus does not grow on it. Pasteurella doesn't grow on it, with the exception of Erogenes. So not all gram-negative rods grow on it either. So it is selective for gram-negative rods, and then it's differential for lactose fermentation. And you might keep wondering, why does he keep repeating this over and over again? Well, let's just say that I have dabbed in teaching, and I have gotten this answer a lot. When I ask which kind of media MacConkey is, I get a lot of the selective for gram-negative. And once again, that's the wrong answer. So the nutrients are in this agar that they need to grow, but what else do they need? Do they need anything else? Yes, right? They need oxygen and carbon dioxide. As you know, most organisms, they're either aerobic, anaerobic, or facultative anaerobes, right? And actually, most of your classic pathogens, they are facultative anaerobes, which means that they grow aerobically, but they can also grow anaerobically. So that's why you have these incubators that have carbon dioxide to give that to the organisms so they can grow. And they also, right, they need the right temperature, right? That's how, we catch infections. Organisms they like our body temperature. So these incubators, they're set, to 35 to 37 degrees Celsius to mimic that temperature. In addition to the temperature, we need the correct pH. And, the books show that most clinical bacteria, they like a range of 6.5 to 7.5. The media has buffers, to make sure to maintain this pH. And they need moisture. So, they need water for certain metabolic pathways. And even though the media has actually a lot of water in it, but when we put it in the CO2 incubators, they start drying. So in order to keep that moisture, you add water to the incubators. So if you're ever wondering, why do I have to put water in the incubator? It's just so you can keep this moisture. So because the organisms need it. And that brings the question, so we have our media, right? And how do we put those samples in the media? Well, it all depends on, depends on the source. Basically, if you have a sore throat and they swab it, if you have a cut and they swab it, so you basically swab one portion of the agar. If you have urine, you stick a loop in the urine sample, and then you put that on the agar. If you have body fluids, right, depending on the amount, you can you centrifuge it and add some of that sediment to it. So it just basically just depends on the source. And then which agar do we choose? You can have a slight variation from lab to lab, but basically they all have the same. It's from the American Society of Microbiology. They create the standards for us. So basically your standard culture has blood, chocolate, MacConkey, and then depending on some sources you add like Martin Lewis if it's a genital source, You tend to add abscesses and other. You add PEA, agar for that. For urines, your standards, blood and MacConkey. For blood cultures, blood, chocolate, and MacConkey, and depending on what you see, you add the extra plates. And then once we have the sample on the plates, what do we do with it? Do we leave it like that? So right, so the answer is no. I mean, it's called So we by adding the sample, we inoculated it, and now we have to streak it. So with a loop, we streak, and then there are two types of streaking. You have your isolation streaking, what they call the 4 quadrant. A lot of hospitals do the 3 quadrant, and then you have the quantitation streaking, which is what the one that you use for your urine cultures. That's for some quantitative bronch washes. So your isolation, right? So basically, you do for the 4 quadrant streak, right? So you see your plate. Imagine, like divided in four. So with your swab, you inoculate your first quadrant. You add that drop of your fluids. And you stick that, that loop in your urine, and you do that, and then you basically start, going from side to side on that first quadrant, and then you turn your plate. That's like about 90 degrees, a little bit less, and then bringing your loop like from your first quadrant, you start making streaking that second quadrant, making sure that at this point in time, the space is not as crowded as in the first quadrant, and you repeat the same process twice. And the goal of this is that at the end, you have some nice isolated colonies. And why do we need that? I mean, you have to keep in mind that our body's full of bacteria. You can have infections with multiple organisms. So if you don't achieve isolation, it's hard to get that organism, by itself. So you can do a proper ID. You can run susceptibilities on it. Also, you might miss an organism. It's very crowded sometimes. Depending on what kind of infection you have, especially if it's contaminated with the based on the source. But you get like a lot of skin flora. Sometimes you have a patients that have these infections like on the foot. With multiple gram-negative rods. So you want to achieve that isolation. It's very important. And then you have that quantitative culture, right? So basically, you use it for urines. You have a loop. The loop, the head of the loop has a diameter of, 0.001 millimeters. So then you actually, you dip your loop in the urine, and then you do one line, one vertical line. And then starting from the top, you go in a zigzagging pattern until you get to the bottom. And basically, depending on each colony, you multiply by 1,000. And the proper format of proper reporting format, it's colony forming units per milliliter. So if you have two colonies, it'll be 2,000 CFU per ml. 10,000 CFU per ml, and that's how you report urine cultures. And then the question is, how do you report your isolation streaking? You know, um, some places do like a 1+, 2+, 3+. Some other places do like occasional, moderate, heavy, depending on the relation to the quadrant. And that's how you will report an isolation culture, which is basically all your other cultures. Like I mentioned before, your quantitation, and you do urines, bronch washes, and then the other cultures you actually streak from isolation. And the same way, if you're trying to have more of a specific organism that you have in your plate, it might be mixed. You're trying to get it by itself. You use that type of streaking, the isolation one. And then so you have your media, you have your sample, you have inoculated your sample, you struck them, they're ready to go to the incubator so they can start growing. Um, Normally you take a look at them after 18 hours, but before you do that, you have to actually prepare a gram stain, right? So this is the process of putting some of that sample on a slide and then, using some staining techniques. You can look for white blood cells and organisms. But that's what we'll be talking about on our next podcast. And with that, my dear audience, we conclude episode 1. You know, thank you everyone for listening, and please continue your passion for what you do. I was actually at a retirement party for a technologist that she worked 45 years in microbiology, having such a great mind, such a great worker. She is my mentor, a friend. I think I learned so much from her, and a fountain of knowledge. And then she said, and this actually stayed with me. She's like, please continue your passion for what you do. Along my career, I had some great experiences, and and some not so great. But it has been a wonderful journey, and there are not that many of us. All the ones in the group who were microbiologists. There are not that many of us that are as passionate for this job anymore. So please, continue. Be passionate. Talk about micro. Find someone that's as passionate as you. I don't know. Try to teach. I think my journey, I also had some great moments. Others not so great. But definitely, it has been a tremendous experience. So keep up that passion. You have a great week. And I will see you next time with another episode of Let's Talk Micro. Thank you, everyone.